Laticifers. Laticifers are derived from the word latex, meaning juice in Latin words. Sometimes called latissiferous cells are the vessels from the Latin word for milk. For example, the lag. Lag means the mill. According to the origin, simple laticifer derived from a single cell or union of cells. Laticifers can be defined as a specialized cell or a row of such cells that secrete the milky fluid, which is known as the latex. The word laticifer is used as a general term to denote the various latex secreting structures. For example, the latex cells, latex vessels, latex ducts, latex tube, and the latissiferous ducts. The latissiferous duct is a cavity into which the latex is secreted. Grouping of the laticifers. On the basis of structures, the laticifers has been grouped into articulated laticifers and non-articulated laticifers. Articulated laticifers are those vessel compounds in origin, consist chains of cells. The walls separating the individual cells either remain intact, become perforated, or are completely removed. Non-articulated laticifers are these are the simple in origin, which through continued growth develop into a tube-like structures. They often much branched, but typically they undergo no fusions with other similar cells. They are simple in origin and typically arise in the primary plant body. Articulated non-anastomosing. These are the compound tubes not connected with each other literally. For example, in plants, Hevea. Articulated anastomosing. In this type of the laticifers, the cells are present in the chains connected with each other literally. For example, in Hevea brasiliensis, Lactuca, Carica papaya, or many hot. The figure shows the articulated type of the laticifers. This is the anastomosing, the connected cells laticifers. This shows that the connection between the two laticifer cells, this is one cell, this is another cell. However, between two cells, there is a connection or a septa is present. Laticifers lying on either side of the xylem strand in a banana leaf that is known as the Musa species. The laticifers are stained red. The term anastomosing implies that the adjacent laticifers fuse with each other to form long secreting structures. The elongated structures are longitudinal sections of the Euphorbia species stem laticifers, which one showing the branch visible as a fork in the form of the fork. This is a branched. The non-articulated anastomosing laticifers characterize all of the Euphorbiaceae families. These are the duct-like structures which are present in the Euphorbiaceae families, basically comprising different types of the latex or the fluid or milky substances. The figure also shows the non-articulated branch laticifers of the Euphorbia species. This figure shows that the embryo, the rectangular, indicates a locus of the origin of the laticifers. The B structure describes the sections to the laticifers showing the multinucleate conditions. The C laticifers branching within the spongy parenchyma as seen in a paradermal section of a leaf. These are the branching pattern of the laticifers present in the paradermal of the leaf. Latex varies in appearance and in composition. The color. The term latex refers to the fluid that can be extracted from a laticifer. Latex may be clear in appearance, for example, in morus, humulus, nerium, oleander. However, most of the Ericaceae family or the milky, for example, in Asclepiasis, Euphorbia, ficus, and Lactuca. It is yellow brown in cannabis and yellow orange in the Papaver. In some species, this is present in the form of light in color or white in color, or sometimes it is also present in the dark brown or red in color. The composition of the laticifers contains various substances in solution and colloidal suspension. For example, the carbohydrates, organic acids, salts, sterols, fats, and mucilages. Among the common components of the latex are the terpenoids, rubber. One of their well-known representatives is the composition of the latex. Many other substances are also found in laticers, such as the cardiac glycosides, in representative of the apocynalis, alkaloids, morphine, codeine, and papaverine in the opium poppy, papaver somniferum, cannabinoids, which is present in the cannabis sativa, sugar is present in the asteraceae, large amounts of the proteins in the ficus calosa, and tannins present in the musa or rhoidae. The crystals of the oxalates and the malate may be abundant in the latex. The starch grains occur in laticifers or some genera of the Euphorbiaceae. The starch grains in Hevea are osteoid, which is the bone shape, whereas those in the Euphorbiaceae assume various forms: rod, spindle, osteoid, discoid, and the intermediate forms, and may become very large. These the photographs shows different types of the starch grains in the non-articulated laticifers of the Euphorbiaceae family. Various types of this is rod shaped, lozenge shaped, or the bone-like structures of the starch grain that is generally observed in the laticifers of the family Euphorbiaceae. A variety of enzymes also occurs in the laticifers, including the proteolytic enzymes, papain in Carica papaya, and lysosomal hydrolases such as the acid phosphatase, acid RNase, and acid protease in Asclepias curicica is also present in the articulated laticifers of the allium. This structure shows that the laticifers are present in the articulated or non-articulated type of the structures in the allium cepa, that is generally known as the onion. When we study the cytology of the laticifers or latex of the plants in different families, this shows that the living protoplast retaining nucleus at functional maturity, the cytoplasm along the periphery surrounding the vascular sac, non-articulated sinusotic condition. If similarity observed in the articulated forms, it is due to the fusion of the protoplast. The latex particles are formed in the cytoplasm. No clear demarcation between the cytoplasm and the vacuole has been observed of such type of the laticifers. The tonoplast break down the escape of the latex particles into the vascular sac, which become part of the latex. However, it means that there is no specific demarcations or the specific type of the membranes are observed or have been observed between the cytoplasm and the vacuoles. That's why the laticifers of various species are difficult to identify. The structure of the wall of the laticifers is also different in different type of the species. The walls of laticifers are non-lignified and the plastic. Generally, throughout the plant body, but most commonly, the phloem. In phloem tissue, these are the present, however, they cannot be differentiated. However, such type of the tissues are generally identified in the form of the non-liquefied the plastic type of the walls are present. They may occur also xylem tissues, for example, in the carici, cortex in musa, pericycle, or on the mesophyll cells also present in the xylem tissues. The walls of the articulated laticifers in the convolvulaceae are suberized. This is also a special type of the substance, which is the polysaccharide. The plasmodesmata, plasmodesmata is the connections between the cells have been observed rarely in the common walls between the laticifers and other cell types. That's why the laticifers don't have such type of the plasmodesmata. Therefore, they also they retain this such type of the milky substances are the latex. That's why they cannot travel from one their canals or their structures to the other type of the cells. This structure shows the articulated and the non-articulated type of the branched structures of the laticifers are present in the cotyledonous structures, early stages of the plant development, whereas this diagram shows the latex cells of the Euphorbia pallidifera, which is a non-articulate. This structure shows the latex vessels of the Carica papaya. However, in case of the resin, this is a resin canals, a resin duct of the pinus, which is the schizogenous type, oil cavity of the citrus fruits, which is the lysogenic type, and the protoxylem lectum of the maize, which is the lysogenic type of the canals are present, which are basically the components absorbing the latex or other types of the substances which are generally known as the secondary metabolites. The development of the latex containing laticifers cells may be simple or branched and is derived from the enlargement of a single cell. The latex vessel is simple or branched tube that usually anastomoses with similar tubes. It is formed as a result of the enlargement and union of the chains of the cells. The latex tube usually means either a latex cell or the latex vessels. The borderline between the different terms of the latex secreting structures, for example, the latex cell, duct, vessel, tube, etc., is ill-defined, and so the word latifier is introduced as a general term. The laticifers may be simple or compound on the basis of the region. The simple latifier is derived from a single cell, whereas the compound laticifers originates from a longitudinal file of the cells. The principal source of the commercial rubber is the bark of the para rubber tree, which is known as the Hevea brasiliensis. Hevea brasiliensis is generally known as a rubber plant. The natural rubber is produced in over 2,500 plant species have been achieved, but few plants contain the enough to make the extraction worthwhile, which is the examples of the Hevea brasiliensis. Among plants of the secondary importance as rubber producers are the Russian dandelion, which is known as the Texcum Hoxae astraceae family, the Syrah rubber tree, Manihot, which belongs to the Euphorbiaceae family, African rubber tree, and the African woody vines, Landolphia, and the Carpodenus. All three Apocynaceae type of the plants are the species. Such type of the plants related to the Apocynaceae. The woody climber, Cryptotegia grandiflora, which belongs to the Asclepiadaceae family, which is native to the Madagascar. Most of the rubber leaden latex obtained from the bark of the Hevea brasiliensis during tapping comes from the layers of the laticifers located in the non-conducting phloem. The conducting phloem is restricted to a narrow band about 0.2 to 1 millimeter wide next to the vascular cambium. However, during the collection of such type of the latex or the rubber from the Hevea brasiliensis, the care should be taken during the tapping into the penetration, the conducting phloem so as to avoid injury to the cambium. If the cambium damages, it means that the plants will also be damaged. So the collection of the laticifers or the latex from the Hevea brasiliensis can be collected uh, from the outer surface of the foil. There is a possible functions of the laticifers or the latex containing the cells in the plant system, which is it is a vital sap vessel and similar to the blood vessels of the animals. Just like that of animals, having the vessels or the arteries, just like that of such type of the system has also been constructed or prepared by the nature in the plant system. It takes part in translocation of the assimilates that is associated with the phloem. It stores food materials. It is now considered as the secretory tissues where the secretory substances do not re-enter the plant metabolism. The laticifers regulate the water balance in plants. It has a role in the transport of oxygen and healing up of the bones as well. That's why generally we have seen that in different types of the plant, for example, in acacia, as well as in the Calotropis procera. Such type of the plants having the latex, ample amount of the latex. When injury comes on the surface of the stems of the leaves, such type of the injury may be healing up in a few moments. It acts as a defense against the herbivores and the microorganisms. The human ocular tissues are damaged by the latex of the Calotropis procera.